Hi everyone. I'm grateful to have this opportunity to present my work here. This is a joint work with my PI, Yoon. We're at University of Pennsylvania and UC Berkeley. Let's begin with the regulation of gene expression. So as everyone knows, DNA transcribes into RNA and then translates into protein. Fortunately, given the development in technology like microarray and RNA-seq, the transcription dynamics has been studied extensively. However, the study of translation dynamics has been a little bit more difficult until recent development of ribosome profiling in 2009 by Ingolia. So what happens in translation? Well, we can roughly decompose into initiation, elongation, and termination. In the initiation, two subunits of the ribosome bind to the 5' end of the transcripts and then traverse this transcript. And at each codon position, the corresponding tRNA comes and then you attach a new amino acid to the newly synthesized peptides. As you can see, this can be fairly complicated, like a resource competition, because you compete for tRNAs, the mRNAs will be competing with each other, and there are many great works that Dr. Tuller has presented, show us in the special session. And what is ribosome profiling? Just to make sure everyone's on the same page, ribosome binds to this mRNA, and if you can immobilize the ribosomes and then you do nucleus digestion, you can retain the small fragments that are protected by the ribosomes and then align into the genome to the, or the coding region, you would get a profile of the distribution of the ribosomes. And this has been shown in Ingolia's first publication that is could potentially be very useful, because he showed that ribosome protein density correlates better than mRNA density with the protein abundance. So yes, this ribosome profiling may tell us something more about this translational dynamics. Well, what I want to do today is looking at these ribosome profile shapes. I wonder if we can use sequence context to predict what the ribosome profile shapes will look like. Why am I interested in this problem? Well, there are many potential applications. For example, isoform-specific ribosome density inference, or some interpretation of the fast and slow codons, and furthermore help us to design in synthetic biology a fast translating transcript. And also if I can do this methodology may be applied to other types of data like RNA polymerase II during transcription. As Dr. Tuller pointed out in a special session, there are many different hypotheses about factors influencing translation, like codon bias, mRNA secondary structure. I think I've heard many great talks about secondary structure in this conference, and also, people found initiation is probably the rate limiting factor, at least in yeast, and maybe the positively charged residuals on nascent peptides could also play a role in this translational dynamics. Though there are many hypotheses, there is one thing in common, that it's transcript sequence dependent. So I think it's reasonable to ask, can I use these sequence context to predict the ribosome profile shapes? Here I'm just, as an illustration, showing one of the examples in yeast, this ribosome profile. One thing to note is that it is very ununiform. So I think it's interesting to ask on which codon these ribosomes sit on, and maybe also what are the neighboring codons. Why I think this may be important? Because the ribosome occupies a space not only just one codon. And also, how, as a researcher starting my career in machine learning and statistical signal processing, I think this is an interesting problem, because we want to use sequence of codons, which is categorical variables, to predict something that is continuous, because we normalize these densities to get the marginal density. And also, I should point out that I'm using publicly available data sets in two treatments, cyclohexamide treated, and also the more recent flash freeze protocol in 2016. These are all publicly available on NCBI. Now, before I talk about how we build a model to do the prediction, I think there is one thing we should ask to ourselves: to what extent can we do the prediction? There may be noise inherent in the data, and we thought this is very like an analogous problem in signal processing, like wavelet analysis. Let's take a look at this picture. It's probably the one with the highest resolution. You can really decompose the signal into global morphology like this one, plus a little bit more details to get into this finer resolution picture. And to be more precise of what I mean, this is like wavelet vector space. So let's assume we represent the density, the ribosome density function as P of X. And then we can represent this signal as a linear combination of the basis functions, phi's and psi's, written down here on this equation. These basis functions are very interesting in wavelet analysis, that they satisfy some nested property. What do I mean by nested property? If you consider the vector space spanned by these bases, they are, which we denote as VM and WMs, they are nested. To illustrate that, let's say we call this the global morphology V0. You see V0 is in the space of V1, and V1 is in V2, V2 is in V3, so on and so forth. And you can also consider the space in between, W0, W1, W2, so on and so forth. So we thought, let's try to decompose our ribosome profile. This is the raw data normalized by the total counts on this transcript, and then into different spaces. So as you can see, it's really projecting this signal onto V0, V1, V2, V3, so on and so forth. As you can see, this is relatively flat, and then you get a little bit more details in V1 and a little bit more in V2, and V7 is, in this example, the closest to the original signal. So by now, we have our signals prepared, that we decompose the signal onto different scales, and we want to know to what extent can we do prediction. The other piece that we're missing is the predictors. So we decided here, we decomposed the codon sequences into really 64 binary sequences. So for example, for codon AAA, it consists of 0 and 1s, these black dots. If at this position it is of codon type AA, it will be 1. Otherwise, at the position for other codon sequences, it will be 0. And then, notice at the beginning I say maybe it's not only on which codon you're sitting on, but also the neighboring codons. So we decide to use a kernel smoothing method to incorporate the neighboring information. And this is written by C tilde, which is a kernel smooth function of CT. This is the kernel function, and what I'm plotting here in the black curves are really using a symmetric Gaussian kernel for illustration. But there's something interesting about this, specifically this translational dynamics, that the ribosomes are moving from the 5' end to the 3' end, and if let's think about a traffic jam, if there's a car in front of you moving slowly, you can't really move forward. So maybe it would make sense to use asymmetric kernel versus symmetric kernel. So we decided to propose a modified gamma distribution. This is gamma distribution. We shifted to such that the peak is located at 0, and we also consider the reflective function here. These are really the way we build the predictors. I'm further illustrating here as a 0-1 binary codon sequence, and then these are missing, but they're really, I'm plotting the kernel smooth function using either symmetric or asymmetric modified gamma distribution, or even the reflected one. And here we use a sparse regression model to do the prediction. So PTS is the ribosome density function PT projected onto subspace S, where S can be V0, V1, or W0s. The vector space you obtain by wavelet analysis. And ZTX is really the predictors we build using the kernel smooth codon sequences. Beta is the regression coefficients. We try to minimize the sum of squares. And we also impose a regularization in penalizing the L1 norm of the beta, the regression coefficients. And this is because, as you see in ZT, we are concatenating the smooth codon sequences at different bandwidths denoted by sigma 1, sigma 2, up to sigma b, where capital B is the number of bandwidths we have considered in this model. And by using more and more bandwidths, definitely the number of predictors increases. Okay, here is one of the example about how we do in the prediction. I am showing cyclohexamide data on the left and flash freeze data on the right. The black curves are the real data and the blue ones are the prediction. So here I'm showing the first 5 scales. As you can see in V0, you see roughly there is a 5' ramp is more severe in flash freeze data. I think this is also discussed in the literature. We can roughly capture that, and let's say we look at the finer scales before. There is a bump here, a few more is here. It looks like our model can also capture that. Similarly, you can take a look at the plots on the right where I'm showing the flash freeze data that shows we can capture the global morphology. And here is a summarized table of the performance. I'm showing the top row are the cyclohexamide, the bottom row are the flash freeze data, and I group the genes according to their length into 5 categories. And some general trends to point out is it looks like overall, yes, the global morphology is easier to predict. The numbers are really the correlation coefficients between the real data and our prediction. Also, it looks like roughly in general, the shorter genes are easier to predict, and this is probably because we're guessing the longer genes may have more complicated secondary structure that we are not including in the model. And another property of this method is that we can check, because we impose a sparsity constraint, so we can check the regression coefficients and see how often these predictors has been selected. And here I'm showing on the top cyclohexamide, on the bottom flash freeze, and it looks like there are a few interesting features, like CCG is selected here in flash freeze. CGA is heavily selected in both cyclohexamide and flash freeze, and CGG is, yeah, it's both in cyclohexamide and flash freeze. We confirm this are some of the slowest codons translating in Liana's paper in 2014, published in eLife. And I also heard many interesting talks from Tuller's lab this time. And also, we think this is interesting because CGA is positively charged, and if you think about the ribosomes, there is a tunnel, and the tunnel is negatively charged, so maybe that's why CGA can slow down the translation. This is another plot showing, I did an enrichment analysis, and I'm showing the p-values versus the selection frequency. This selection frequency refers to how often a feature in the regression model has been selected. This enrichment analysis is done in a way that, so I check those regions within a window size, those regions that you have higher density of ribosomes, and then I find those, and I compare it to the average distribution of the codons and see if that codons is enriched in those high density regions. So probably as expected, those red ones are codons that are enriched and also selected with high frequency. And the blue ones are not enriched, not oftenly selected. The green ones are interesting. They are not enriched, but they are selected frequently. This is probably the reverse, that they are depleted in these high density regions, and so it's also useful for the model to include them as a predictor. Also, it looks like Flash freeze data gave us cleaner results than the cyclohexamide, where we don't have these yellow codons that are enriched but not selected with high frequency. So by that, I conclude my presentation. This is the results conducted on yeast, and we show that for the global morphology, it looks like we can predict the shapes of the ribosome profile with pretty good accuracy. Some interesting applications I mentioned at the beginning is isoform specific ribosome density inference. This is because, notice, if you have a ribosome sitting on the exon that is shared among many isoforms, it will be hard to locate that isoform to the correct isoform because ribosome footprints are fairly short. However, using our model, if we can do the prediction of the ribosome profiles, we can statistically assign these ribosomes to hopefully the correct isoform. In the regression coefficients, I have shown that there are some biological interpretations of fast and slow codons, and hopefully in synthetic biology, this will guide us for design of the transcripts. And also our RNA polymerase II is another type of interesting data that may be applicable for us. So by that, I conclude my presentation, and I just want to acknowledge my PI Yoon Song and all the lab members, and my funding resource, NSF and Simons Foundation, and also thanks to the conference for the travel fellowship. Thank you.